All right, this is part 2 in the video review for exam 3. Part 1 covered viruses, so I'm going to pick up now with eukaryotes. My plan for this is fairly similar to the way that I did viruses. I'm going to go through conceptual stuff, some of the specific pathogens that we went into detail on, but those that we just went over superficially and the information's just in text or in the notes, I'm not going to spend time on. So first thing, you should be able to sort of make a comparison between eukaryotes, bacteria, and viruses. And I know the bacterial stuff is a carryover from last time, but it's pretty straightforward. And the contrast is they differ greatly in their strategy. And the two things that you can sort of compare, one is specialization. Eukaryotes, as far as parasites go, are about as specialized as you can get. They are complex cells, they have organelles, a lot of times they're motile, they have flagella. In the case of worms, some of which aren't even microscopic, they can actually burrow through skin to get inside the host. And so that specialization allows them to do a lot of things. One is they're adaptable and they can live in a lot of different environments. That might mean different organs within the host. It might mean a wide variety of different animal species, not just humans, but other mammals. And it might mean that they can live in other environments like soil or on surfaces as well. And so it gives them a big advantage in that way. The downside is being complicated means it takes more time to replicate. You've got more organelles to copy, you've got bigger cells to duplicate, you've got higher ATP requirement. What this translates to clinically is a lot of times viruses and bacteria have pretty short preclinical periods, meaning you're exposed to the bacterium or the virus and within 1, 2, 3, 4 days, usually the symptoms are coming on pretty strong. For eukaryotes, that amount of time can be quite a bit longer because it takes them longer to get going. The advantage for the host is it gives you a bit more time to detect them as far as the immune system goes. So just be able to put these on a continuum of complexity. Viruses are as simple as it gets. The eukaryotes that we'll talk about are on the complex side. Bacteria, somewhere in the middle. You could also, I suppose, lump prions in there, which are even more simple than viruses, but they're sort of strange in that they don't even replicate like a normal parasite does. Okay. A few other things to get out of the way. The term host, you're very familiar with. So for a viral or bacterial infection, if we're talking humans, obviously the patient is the host. Definitive and intermediate, there are some eukaryotes that sort of jump back and forth between animals and humans. We're not really talking about any of those in detail. I do want you to be able to differentiate between these two. Zoonosis is when a parasite, a virus, a bacterium, a eukaryote, whatever, is transferred from a different animal to humans. Easy example, swine flu, bird flu. If a human eats contaminated meat that has from a cow that had BSE, mad cow disease, those all count. The reason that's called zoonosis is the animal was actually infected and they had symptoms. And so when a pig contracts influenza, they have a respiratory infection, they have the flu. And so it's an actual transfer from one to the other. A vector is something different. This is like a mosquito with malaria or ticks with Lyme disease. The insect doesn't actually become infected. So mosquitoes don't have symptoms from malaria. The mosquito is not trying to actually cause damage. It just transfers it from one person to another. And so a vector is really just a carrier. Zoonosis is when other animals actually get the infection and experience symptoms. Okay, last general thing to clear up is terminology about naming sort of categories of eukaryotes. The one that we're going to spend the most time on, the group is technically, the big broad group is protists. The two subgroups are algae and protozoans. We don't care about algae for this class because they don't infect people. They're important for ecological reasons, but not for medicine. Protozoans is probably the name that we should use, but almost everyone just says protists. And so for the sake of this class and for your future most likely, you can probably use these two names interchangeably, and most people do, so it's not a big deal. And so I usually say protist because that's the common terminology, but the more specific protozoan would also be fine too. Great. So the protist that we spent the most time going into detail on is Plasmodium, and there are a few different species of Plasmodium, Falciparum and Vivax, that are the causative parasites in malaria. So malaria is the disease and the symptoms. Plasmodium is the genus, and obviously these are the species that actually cause the infection. The mosquito, Anopheles, is the vector. So again, Plasmodium doesn't infect mosquitoes. They just get carried around when a mosquito transfers blood from one person to another. And malaria is by far the most common protozoan disease in the world, probably more common than every other protozoan disease put together. And because of that, we're going to spend some time going into details. This reinforces what we just talked about with protists being more complicated and taking longer to replicate. Incubation period, meaning from the time the infected mosquito bites you until you start experiencing symptoms, an average time is close to a couple weeks. The reason for that, like I said, it takes longer for them to build up numbers. Bacteria, go through a replication cycle every 20 or 30 minutes. For protozoans, it can be several hours. And so that really strings along the amount of time that it takes to get the infection to a point where it causes symptoms. And I want you to be able to differentiate the symptoms and where they're coming from. So I'll come back to this in just a minute. But for right now, these first series of symptoms, which is basically fatigue and nausea and sort of a general achiness, is different than these repeated bouts of chills and fever that happen every 2 or 3 days. And if you look at the life cycle of Plasmodium, you see it goes through 2 major phases inside the host. The first phase, immediately after you're exposed, and we're not going to worry about names, sporozyte and merozyte and gametophyte, those are zoology terms. What I want you to know is stage 1 in all this, exoerythrocytic just means outside of red blood cells. But the place that it's actually happening is in hepatocytes of the liver. And the way this works is a very small number of cells. When a mosquito bites a person, the little bit of sort of backwash that the mosquito injects into your blood before it takes the blood draw, really, really, really small. And so not many cells actually get in your blood. What they're going to do is start infecting liver tissue, but since there aren't very many cells, right, it takes a long time for them to replicate, hence the 10 to 14 day incubation period. Once they have replicated enough, the hepatocytes, the liver cells that are infected, will rupture. And at that point, the Plasmodium changes to a different stage in its life cycle and now becomes receptive to infecting red blood cells. So that's the erythrocytic phase. Red blood cells are erythrocytes. And at that point, right when the liver cells start to rupture, that corresponds with those initial, first detected symptoms of fatigue and nausea from liver dysfunction. When red blood cells get infected, this continues as a cycle, right? Infect a red blood cell, multiply and multiply until you have enough Plasmodium that they rupture. It's the rupturing right here that causes symptoms. That cycle takes 2 to 3 days. So now you have the next part of this, right? Fatigue and nausea once the liver infection gets bad enough that cells start to rupture. Bouts of chills and fever and sweating is from the systemic inflammation that comes from red blood cells rupturing. Okay, at that point, obviously, if another mosquito bites that person, now they're taking blood out when red blood cells are infected with Plasmodium, and of course they can fly away, bite another person, and transfer. And the reason that vector strategy is so effective is, obviously, Plasmodium is just sort of hitching a ride on what mosquitoes already want to do very badly, which is draw blood from people, because that's what they feed off of. Okay? So this all explains the symptoms of malaria, and the vector strategy shows you why there are such an enormous number of cases, hundreds of millions every year. All right, so you've got a malaria infection. Sometimes this just resolves itself. Sometimes the symptoms can get quite bad. Some strains and subspecies of malaria can cause cytoadherence, and all that means is, it really translates to cell stickiness. And if red blood cells start getting sticky, this produces a really similar effect to clotting. It's not technically clotting because there aren't platelets involved, but it has the same effect. Blood starts to get thick and viscous, it doesn't flow very well, and organs that are very sensitive to oxygen deprivation can get in trouble. Obviously, the brain is one of those. If oxygen levels get too low, it can result in coma. And the other major concern is during pregnancy, there are a lot of blood vessels at the chorionic villi where the fetal blood supply meets the maternal blood supply at the placenta. If the fetus isn't getting enough blood flow, obviously it's not getting enough nutrients. And so either premature birth, miscarriage, or if nothing else, very low birth weight from lack of nutrition can be serious complications. Right. The next few, we went through entamoeba and Chagas disease and toxoplasmosis. These are laid out very clearly on the slides. I'm not going to read off the notes to you. So be familiar with the very general ideas of transmission and symptoms and so forth. For parasitic worms, I'll give you a quick overview. What I want you to be familiar with each is generally mode of transmission and symptoms. Worm infections, roundworms, hookworms, etc. in the U.S. are, they're relatively common. They happen. They're fairly easy to treat. And so it's not a big deal. But just take a look at the life cycle. Be generally familiar with each. For example, roundworms are taken in through the oral route, whereas hookworms have teeth, obviously, that can burrow through your skin. And so just be familiar with how they actually get inside of you. And let's see. For filariasis, just give yourself a quick refresher on immune system, or lymphatic system, and why they cause this effect, blocking lymphatic tissue, blocking fluid flow. And for tapeworms, we didn't go into any specifics about the different types. Those names don't really mean anything anyway. And the symptoms are so mild on tapeworm. All they really do is steal food from people. There's not a huge amount of information there. Okay, for fungi, we're not doing a bunch of basic biology, anatomy and genetics on fungi, but I want you to know a couple things. First, that they're completely different as far as classification goes from protozoans, and fungi come in a lot of different forms. Everything from single-celled yeasts to molds to mushrooms to a lot of different variety. And we don't care about the vast majority of those. You've seen some of these growing on your plates in class, and if you zoom in on them under a microscope, you see these long filaments. Right? Those are hyphae. They look like this. They're essentially sort of pseudocellular compartments that have little spaces between them. And so it's kind of like one big continuous organism. The cells aren't completely blocked off from each other, but we think of it as a multicellular organism. These don't really infect people very much. And so you'll see those on your growth plates in class. They're very, very common in the environment, very common in soil, but we don't have to worry about them quite so much. We do have to worry quite a bit about single-celled yeast. They're not the cause of many really serious infections, but they cause nagging problems. And the thing that makes yeast different from protists, they can switch life cycles. And that's not a huge deal clinically, but it kind of gives yeast an advantage in some ways. And all it really means is, instead of being like bacteria where they're always just dividing through binary fission, haploid all the time, never really doing any sort of sexual reproduction, yeast can switch back and forth. So they can stay in these haploid phases. This is very much like bacteria. Just divide, 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 all by themselves. But if conditions are right, which usually means they have plenty of nutrients available, they can start doing something called conjugation. And that's basically just a sort of primitive form of sexual reproduction where two different cells, they actually have something that's kind of like a gender, an alpha and an A, which I know is confusing, have to match up with each other. They can become diploid, meaning that the cell now has two copies of its chromosome, not just one, like it would in the haploid stage. And then you can get some mixing of genes. At that point, eventually, yeast can go back to a haploid state again. And all that really does is it gives some genetic variation. And so they have the ability to do some of the things that bacteria can, which allows them to divide quickly in the haploid state, and some other things, this conjugation step, which is what other eukaryotes do, which gives them some genetic variety. Right. We didn't spend time going into a bunch of detail on any specific fungi. What I would like you to know is the vast majority of yeast exist as normal skin flora. Most of them are commensal, meaning there's really no established benefit to having yeast growing on your skin, but they're really not harmful. The vast majority of the time, they just live there, not bothering people, and for the most part, feeding off of keratin. So they're called dermatophytes for that reason. Problems can come about when you get certain species that in particular situations can become mildly pathogenic. Ringworm, I would like you to know that ringworm has nothing to do with the worm. It initially was thought to be a small microscopic worm coiled up in your skin because of the round structure of the lesions. But it's just the shape that the fungal growth takes. And that's very similar, closely related to athlete's foot and tinea cruris, which are both fungal infections, and they just overgrow when there's an excessive amount of moisture for a long period of time, and they replicate more than they otherwise would. All right, another that I'll briefly mention, aflatoxins are produced by a group of fungi, genus is Aspergillus, and just a reminder for this one, because it might not be obvious just reading over it. The fungus doesn't really infect people. It's incredibly rare. The toxins are produced when the fungus reproduces, usually on crops. And we talked a little bit about its role in liver cancer if you're exposed to even moderate amounts. And I showed you this data showing you that aflatoxin can lead to stunted growth. And so almost everything that you eat from a grocery store that contains peanuts or corn has at least trace amounts of aflatoxin. And if children, very small children especially, consume really, really large quantities of those two foods, they can eventually sort of get above the limit because the individual food might come below the threshold. But if you consume lots of it over and over again, especially since aflatoxins bind to DNA and stick pretty tightly, it's something that can accumulate in the liver. And so the FDA monitors levels very, very closely to make sure they don't get too high. Okay, for the prion section, we spent quite a bit of time having a discussion about this in class, and I'm not going to go into that level of detail, but I am going to summarize the most important information that I want you to know for the exam. And the first thing is, I'll use this slide to do it. I want you to have a good explanation of, one, what makes prions so strange. And the thing that made them so strange and difficult to investigate and difficult for a lot of people to accept that they were even real is that they have no nucleic acids. There's no DNA, there's no RNA. That means they can't make copies of themselves. And a question that you should be ready to answer on the exam is, if prions can't really make copies of themselves, how can they replicate? And the answer, which again I know we've spent quite a bit of time going over, so I won't belabor it, but the idea is prion form of protein is exposed to normal, healthy, correctly folded version of protein, blue square or cube. And when the prion version contacts the normal version, much like an enzyme, it can cause a shift and create a misfolding in what was a perfectly healthy protein, which can now spread to more and more and more of those proteins. The catch is, unlike bacteria and viruses, in order for a prion to spread, Okay? And this first model, what they call the illustrator calls it the refolding model. I like to call this the exposure model. The idea is this healthy version of the protein, the normal, correctly folded version, has to be present inside the host. Okay? And so if you're exposed to a prion, if you didn't have this, the correctly folded version, there would be nothing to convert, right? Nothing to change. The prion would eventually get degraded and go away, and it wouldn't be an issue. And so be able to put that into a description if I ask it, on a written question. Explaining that they don't really copy themselves, but they take something that the host already has, that correctly folded version of the protein, and convert it into something pathogenic, the prion version of the protein. Okay. So that explains how something can be contagious without actually truly replicating itself. The catch is this model works very well for something like kuru. Right? You eat someone's brain that had kuru, the prion molecules from that brain tissue get it inside your brain eventually, probably through the GI tract and the nerves that innervate that tissue. And then eventually it spreads far enough and you have the disease yourself. With something like Alzheimer's disease, which we talked about in class, acts like a prion in many ways. That's called the seeding model. I prefer to call it the spontaneous model, meaning Alzheimer's isn't contagious, at least not right now, because we don't expose ourselves to Alzheimer's brain tissue. And so the idea is somehow there has to be a spontaneous conversion from the healthy version of the protein, before you have Alzheimer's disease, into the prion form. And once you've started making some of those prion forms of the protein, then it can start spreading in the exact same way that happened up here. And so the concept is the same. A misfolded protein causes other proteins to misfold. The difference is sometimes it's contagious, like in kuru. Sometimes it's spontaneous, like in Alzheimer's disease. But just because Alzheimer's isn't contagious now doesn't mean it couldn't be. It's just that we don't eat people's brains. And if that happened, it's pretty certain that Alzheimer's would be contagious in that way. It certainly is in mice when that happens anyway. Okay, so the kuru part of this I'm going to let go because I'm not an expert. Alzheimer's disease, however, is something that I've spent a lot of time working on, and so I'm going to use it as an example. The interesting thing here is you've got two separate proteins, amyloid beta and tau. Amyloid beta is usually just abbreviated A beta. The strange thing is Alzheimer's is much, much more common than any other form of dementia. It's actually more common than all the others combined. And the weird thing is most other forms of dementia just involve one protein, sort of clumping and misfolding and aggregating. Diseases like Huntington's and Parkinson's and so forth. In Alzheimer's, you have two separate proteins. Amyloid forms plaques. Right, these orange clumps, stained orange, form big extracellular plaques outside of cells. Tau, a completely different protein, starts aggregating inside of neurons. That's why these tau tangles, they're called neurofibrillary tangles, they look like neurons because they're essentially the dead skeletons of neurons that died when tau started building up and aggregating inside of them. The weird thing about this is it's really strange for a disease that's more common than others to have two separate proteins that are going wrong. It just doesn't make sense. What are the odds that they both start having prion-like activity at the same time just by coincidence? And so the much more likely explanation is not that they both happen separately, but that one causes the other. And there's pretty good evidence now that amyloid sort of gets things rolling. That's the initial starting process. And in some people, but not everyone, that have amyloid plaques building up, they eventually have problems with tau too. And since tau is what actually builds up inside of neurons and kills them, there's sort of a consensus developing in the Alzheimer's research community that clinical trials, which have always focused on getting rid of plaque, haven't really worked. They do get rid of plaques somewhat, but they don't really help patients. Part of that is because tau's already killed all these neurons. There's nothing really left to save. And so if you want to stop Alzheimer's disease, we're going to have to figure out a way to prevent amyloid from initiating that tau pathology in the first place, which is probably going to be very difficult if it's even possible. Okay, this slide is showing you something that's not really a big deal for the test, but just as background. Amyloid is actually a little tiny piece of protein that's cut out of this much larger protein, APP, amyloid precursor protein, easy enough. And this entire coiled structure is supposed to show you APP, and this little piece that gets cut out of the middle, that's A beta. It's that little tiny chunk of peptide that can start aggregating and form those enormous plaques. Since it's such a small little molecule, it takes millions and millions of amyloid molecules to make a single plaque. So, and in order to get it, you have to cut APP with 2 different enzymes. That's not really a big deal, but it's interesting in that people that have mutations in, for example, this beta secretase enzyme that make it overactive, that cuts even more, they have familial Alzheimer's that starts in their 40s and 50s, much, much earlier than normally occurs. And so there are genetic mutations that can speed up the process, very unfortunately. So what that means is you have all these little amyloid peptides. Each one of these squares is supposed to show you a single amyloid molecule. They're floating around by themselves, and over time they'll start building up into 2, 3, 4, 5, 10, 20, 100, 1,000, millions, hundreds of millions when you get to plaques. And so you start off with these single peptides floating around, and over time they eventually turn into oligomers, which just means little clusters of 2, 3, 4, 5, etc. Peptides together, into big fibrils, into plaques, into the giant plaques that we see when you put Alzheimer's brain tissue under a microscope. A couple questions that pop up. One is, where along here is it actually dangerous or toxic? And the answer seems to clearly be this oligomer stage, when you have very, very tiny clusters of 2, 3, 4, up through 15 or 20 peptides together, seem to be very harmful to cells. They're pretty toxic to neurons and culture when you expose them. Plaques don't really seem to do much. And so when we look at pictures like this, it seems like what we're really seeing is the sort of aftermath of it. You're not seeing the process of cells dying. You're seeing what the city looks like after it's been bombed. You didn't actually see the war. And that means we may be going after the wrong thing. Getting rid of plaques probably isn't the answer. You're going to have to target the process much earlier on. The assumption's always been that this process takes a long time. To get from monomers to dimers to trimers to tetramers to on up to fibrils and plaques, it was thought, well, Alzheimer's takes 10, 15, 20 years to start developing and progressing and see symptoms, and so the plaque formation probably takes that long. This experiment showed that that's really probably not the case. They imaged a live brain of a mouse that was exposed to Alzheimer's brain tissue, and so it started forming its own plaques. And what they found is that these plaques, you can see them here in blue, red as blood vessels and green as neurons, those plaques formed quickly. When they looked at that live mouse brain and they measured plaque size every day, what they found pretty much across the board is plaques grew very quickly for a day or two, and then their size stabilized. That's really strange. If Alzheimer's takes years and years and years to develop, how can plaques form that quickly? And then they don't get any bigger. There must be something strange going on. And the idea is, and there's good evidence for this, it was once thought that plaques started off very, very small, all across the brain, kind of like this, and very slowly over time, plaques would get bigger. They get a little bit bigger, they get a little bit bigger, and eventually they would get big enough that they would start causing damage. Obviously, I'm drawing them on here much bigger than they would actually be, but I don't have a microscopic image. This data suggests that instead, plaques probably start off in a very specific location. And from there, they'll spread to the next area, just a few cells over, spread to the next area. And this process of spreading can take a really long time. You might only form a couple new plaques every day. Since these are microscopic structures, it's going to take a very long time to radiate outward, right, all the way throughout the brain. And so the idea is each individual plaque might form very quickly, but it takes a really long time for that plaque formation to spread from the center of the brain, which is where it starts, all the way out to the periphery of the cortex. And so the way that I think that we should approach this is, right now Alzheimer's isn't contagious from one person to the other just because we don't do things to make it contagious. But it clearly is contagious from one area of the brain to another. And so it doesn't spread from person to person, it spreads within one person's brain. And that to me qualifies it as a prion disease. The exposure model was shown in mice. This is brain tissue from a mouse that was exposed to human Alzheimer's tissue sample. This was control. The black spots are plaque. It's pretty clear that if your brain is exposed to Alzheimer's brain tissue, it will spread and infect your brain in a very prion-like manner. Okay. The information here in the slides, starting with this pyroglutaminated information, we'll go over that in class. That's my research. I think it's interesting because I did it, I guess. But it's not critical for the test, and so I'm going to leave it out of the video. The thing that I'll finish with is, if what I'm saying is, getting rid of plaques and tangles after they've already formed, it doesn't work. Brain cells are already dead. They can't be regenerated. There's really no hope for it. The only thing that you can do is maybe try to prevent Alzheimer's. And if you're going to prevent it for a reasonable clinical trial, you have to have an idea of who's going to get Alzheimer's. There has to be a prediction. Okay? And so is there any way that we could predict Alzheimer's? Because we don't want to just go give experimental drugs to millions of random people. Most of them aren't going to get Alzheimer's anyway, and you have no idea what the side effects are. And so what these researchers did is they took CSF samples from patients who had a CSF draw done for some other reason, and they saved the extra. And what they did is they measured dozens and dozens and dozens of different markers. The two that they settled on that were the best predictors, one was free-floating CSF levels of A-beta. The other was levels of a very specific version of tau, phosphorylated amino acid number 181, which isn't important for you to know. But the point is, they then followed these people for nearly 15 years. And what they found was, obviously some people were still healthy and normal. Some people had what's called mild cognitive impairment, which means their brain function isn't what it should be, but they weren't actually diagnosed with Alzheimer's yet. And then the third group, which was diagnosed with Alzheimer's. And what you can see is, if you fell into this area that they have highlighted as red, it's not a guarantee that you're going to be in the MCI of the Alzheimer's group, but it's pretty likely. And if you fall into the green circle, it's not a guarantee that you're going to be in the normal or the MCI group, but it's pretty likely. There are very few people in the Alzheimer's group in that category, and there aren't that many people in the red group in the normal category. And so it's not a perfect prediction. It doesn't tell you exactly for sure who's going to get Alzheimer's and who's not, but it lets you predict this person is at least a lot more likely to get it, this person's a lot less likely to get it. And so the idea is, if we can come up with drugs that there's a decent confidence might work, instead of giving them to people randomly, give them to people that are, in this high-risk area. The other thing is, it's also possible in 5 or 10 years, if they follow up again, because this is a fairly recent publication, some of these people could have moved to there, and some of these people could have moved to there. And so it's sort of yet to be determined. The model might be even better than what these data are showing. Okay? The downside is, if you're going to prevent Alzheimer's, you're probably going to have to use strategies that are, like I said, 10, 15, 20 years away from pathology. That leaves open a huge window for possible side effects. And so this is going to be difficult no matter what. It's a very difficult disease to treat. For the purposes of the exam, focus on how amyloid and tau are acting like prions. Give some of the evidence for that. How does it spread throughout a person's brain? And then briefly be able to describe this experiment and what its significance is.